Hello girls, I hope you're staying safe at home. To continue with the circulatory system, today we are going to talk about white blood cells, abbreviated as WBC, and they are also known as leukocytes. Leuko means white. And they are different from red blood cells because they have a nucleus and they do not contain any hemoglobin. And their number is also much less. It is only about 4,000 to 8,000 per millimeter cubic of blood. Okay. Most of the white blood cells are amoeboid, means they do not have a fixed shape. They can change their shape from one form to another form, and they produce pseudopodia. Pseudo means false, podia means feet. Okay, so with the help of pseudopodia, they can squeeze through the walls of the capillaries into the tissues. This process is known as diapedesis. So what is diapedesis? It is the squeezing of the white blood cells through the walls of the capillaries with the help of pseudopodia. So you can see here how this is the white blood cells. It has produced pseudopodia or false feet, and slowly it will just move out from the blood vessels, and this process by which white blood cells moves out or squeezes out from the blood vessel with the help of pseudopodia, it is known as diapedesis. Now, based on shape and other characteristics, the white blood cells are classified into two categories or two types, that is granular and non-granular, and 5 distinct types. So the different types of white blood cells are granular and non-granular. So when we talk about granular white blood cells, they are called granular. Why? Because their cytoplasm contains granules. It is grain-like, okay? And nucleus is usually constricted into lobes. So here you can see in this diagram the nucleus, 1, 2, 3 lobes, okay? The first type of white blood cell is known as neutrophil. About 62% of the total white blood cells are neutrophils, and they have constricted nucleus. Okay, it is divided into lobes. They usually have 3 to 4 lobes, and cytoplasm is grain-like granular, and they stain with neutral dyes. So dye may be acidic, basic, or neutral. Okay, so when in the lab, if you want to look at a white blood cell, you need to stain it. So the nucleus stains with neutral dyes. That is why it is known as neutrophil. The function is it engulfs bacteria, which is known as phagocytosis. The process by which white blood cells engulf bacteria is known as phagocytosis. They are formed in the bone marrow. Next type of granular white blood cell is eosinophils. About 2.3% of the total white blood cells in our body are eosinophils, and count increases in allergies. Supposing you get some allergies and you take a blood cells, there will be more eosinophils in your body. Okay, so if you look at the nucleus, how many lobes do you see here? 1, 2. So nucleus has 2 lobes. It has large cytoplasmic granules. Okay, the grains you can see, they are very large. Okay, and they stain dark red with eosin, which is an acidic dye. Okay, that is why they are known as eosinophils. Eosin is the name of the acid dye. Okay, the function is it engulfs bacteria, again phagocytosis, and it secretes antitoxins, and it is associated with allergy. That is why the count increases when you have allergy, and again it is produced in the bone marrow. Okay, the third type is known as basophils. About 0.4% of the total white blood cell are basophils. Okay, now if you look at the nucleus here, you do not see how many lobes are present. So it is indistinctly lobed. Nucleus is large, looks very big. Okay, and granules stain with basic dyes. That is why they are known as basophils. Okay, especially methylene blue. Now, the function is it releases chemicals or histamine for inflammation, which dilates the blood vessels. Okay, and it is also produced in the bone marrow. Now, moving on to non-granular white blood cells. Okay, so non-granular means cytoplasm does not have any granules. It is not grain like. They are granulocytes. Okay, they have a single large nucleus. It is not lobed like the granular white blood cells. So there are two types: lymphocytes, which constitute about 30% of the total white blood cells in our body. Remember, lymphocytes are the smallest of white blood cells. So if you are to name the smallest white blood cells, what will you name? Lymphocytes. It has a single large nucleus, as you can see here, a single large nucleus. And what is the function? It produces antibodies. Okay, and where is it formed? It is formed in the bone marrow and lymph glands like spleen and tonsils. Okay, the last type of white blood cells is also non-granular, and they are known as monocytes. About 5.3% of the total white blood cells are monocytes in our body. Okay, here can you see the nucleus is kidney shaped? Okay, so it is large, it is kidney shaped, the nucleus is large and kidney shaped, and at the site of infection it transforms into macrophages to to ingest the germs. So what is the function? It ingests germs. It, like you can say, it gobbles up the germs. It is formed in the bone marrow. So moving on to the origin and life of white blood cells. I already told you when we discussed the different types of white blood cells that they are mostly produced in the bone marrow. So they are produced in the red bone marrow, lymph nodes, and sometimes even in the liver and the spleen. Okay, and they live for about 2 weeks. The neutrophils live only for a few hours, and about 125 billion neutrophils are produced every day. The old and worn out white blood cells are destroyed in the same manner as the red blood cells, that is in the liver. Okay, remember, leukemia is a cancer of the tissue forming white blood cells whose number increases at the cost of red blood cells. It is a fatal disease. Currently, the only treatment is blood transfusion. And leukopenia is the abnormal decrease in the number of white blood cells. Okay. Now, moving on to the functions of white blood cells or leukocytes, its main function is to defend the body against germs. Okay, first is phagocytosis. I already told you the process by which white blood cells engulf bacteria is known as phagocytosis. So it is a process in which most white blood cells and mainly the neutrophils engulf particle-like solid substances, especially bacteria. So this is a defensive mechanism against germs. Okay, and abnormal increase in the count of white blood cells up to 50,000 or more per cubic millimeter indicates that some infection is there in the body. That is why whenever you fall sick, whenever you have some infection, you are asked to take a blood test. So they are taking a blood test to count the number of white blood cells. So if the white blood cells have increased, that means you have some sort of an infection. Okay, inflammation or swelling up of certain regions, like when a mosquito bites you, you'll see a small inflammation, a small part of your skin is swollen up. Okay, so it occurs due to the reaction of tissues to injury or to localized invasion of germs. The inflamed spot has different characteristic: local heat, it becomes red, there is swelling, there is pain, sometimes it itches. Okay, so here at this point the leukocytes, especially the monocytes and neutrophils, migrate through the walls of the blood vessels by diapedesis. That is, these white blood cells move out from the blood vessels and come to that point wherever there has been an entry of bacteria, and they fight against the disease causing germs. They also destroy the damaged cells by phagocytosis, that is engulfing, eating away the damaged cells. Okay, and sometimes when at that point you will get pus, whitish fluid which accumulates there. That pus is made up of dead white blood cells together with the tissue cells destroyed by the bacteria. Okay, the next function of white blood cell is formation of antibodies. The white blood cells, especially the lymphocytes, produce antibodies which kill or neutralize the germs. Okay, any germs that has entered our body is a foreign body, so lymphocytes produce antibodies to act against that foreign body so that it is neutralized or it is killed. Okay, and sometimes the bacteria, they also produce poison, and even this poison is neutralized. And introducing weakened germs or germ substances, these are vaccines. During vaccination stimulates formation of particular antibodies which at a later period will destroy the particular disease causing germs if they enter into the body. That is, when you go to get yourself vaccinated, I think when you were little, when you were small, you got vaccination for various diseases. So actually what is being introduced into your body? So the germ, the very germ which causes that disease. Supposing you got vaccinated for measles, what is being put inside your body? The germ or the bacteria which causes measles is being put inside your body. So as soon as that bacteria enters the body, what start acting against it? The white blood cells. So they will produce antibodies, and then they will know in future, if the same germ enters your body, they will know what type of antibodies are to be produced so that that certain germ is killed. When germs which cause diseases enter into our bloodstream, they produce poisonous substances called toxins. Toxins means poison. So after they enter our bloodstream, the lymphocytes of the blood, they produce a chemical substance called antibodies which start circulating freely in the blood plasma, that is the liquid part of blood. So the antibodies are specific to the toxins for which they are produced. So this antibody will only act against the certain disease for which it was produced. Okay, so the antibodies, they act against the toxins, so they are antitoxins. They neutralize the poisonous effect of the toxin. If we take, for example, the COVID-19 pandemic right now, you must have heard, the government wants to make a plasma bank. Just like we have a blood bank, a plasma bank is being thought about by the government. That is, people who have been cured of the coronavirus can donate their plasma, and it will be kept in banks and that will be injected into the body of new patients. Okay, and the plasma of the already cured patient contains antibodies for coronavirus, and then that antibody will be able to help the new patient who is suffering from the disease. Okay, now antibodies may persist long after the disease has been overcome. That is why the government is asking patients who have been cured of coronavirus to donate their plasma, because the antibodies will remain in their plasma for a long time, and the person who has recovered becomes immune to the disease. If the coronavirus again enters that person, what will happen? That person will already have the antibodies, and the antibodies will act against the virus. So this is the underlying principle of vaccination. Also, vaccination, I already told you that what is injected inside the body, bacteria, the germ for that particular disease is injected into the body, and the body will start producing the antibodies. And later in life, if the same bacteria or virus attacks the body, then the body is ready to produce the same type of antibodies. Okay, now next we move on to blood platelets. Okay, blood platelets are also known as thrombocytes. They are the initiators of clotting of blood. Initiates means they start the clotting of blood. Now, blood platelets are minute oval or round structures. They do not have any nucleus. They are floating in the blood. There are about 2 lakh to 4 lakh per cubic millimeter of blood in an adult. The platelets are, they break off from some giant cells called megakaryocytes in the red bone marrow. And these are budded off from the megakaryocytes, like they are broken off, like they break off, and each one is completely surrounded by a membrane. So it is oval in structure, round in structure, okay, and they live only for 3 to 5 days, and they are destroyed in the spleen. The spleen, it is a lymphatic organ, and you find it in the abdomen, and they are very important in clotting of blood. When you cut yourself, you will bleed for some time, then after some time you see a clot being formed. So it is the blood platelets which help in clotting of blood, okay. Now at the site, now you have cut yourself, so what happens wherever you have cut yourself? The platelets disintegrate to release a chemical substance. So what happens? The platelets will break up into small parts, and they produce a chemical substance which is known as thrombokinase, which initiates or starts the process of clotting of blood, okay. Now, the function of the platelets, as I already told you, the main function is clotting of blood, also known as coagulation, okay. So when a blood vessel is cut, the blood escapes from it, you all have seen, and after some time a clot is formed on the wound and the flow of blood is stopped. That is the main reason, to stop the flow of blood, okay. And if not, what would happen? There would be continuous bleeding, and the person would die. So clotting is also known as coagulation, and this is how it takes place. Now the injured tissue cells and the platelets disintegrate at the site of the wound, and they release a chemical substance. I already told you, thrombokinase, also known as thromboplastin, and most recently it is known as factor X or Stuart factor. So it has 4 names, okay. Thrombokinase, thromboplastin, factor X, and Stuart factor. Now the thrombokinase has been released at the point where you have cut yourself. Now this thrombokinase, it acts as an enzyme, okay, and with the help of calcium ions present in the blood plasma, it changes prothrombin, which is inactive. It is there in the blood, it is inactive in the plasma of the blood, into thrombin. So now it becomes active. That means it has work to do. It has woken up, you can say. It was sleeping. Now the thrombin, it has become active in the presence of calcium ions, reacts with the soluble fibrinogen of the plasma. So in the liquid part of blood, there is a substance which is known as fibrinogen, and it is dissolved in the plasma, okay. So what does the thrombin do? With the help of calcium ions, it reacts with the soluble fibrinogen, and then it converts it into insoluble fibrin. Insoluble means it is solid. So fibrin is a solid substance which form long thread-like structures. So these microscopic threads of fibrin are sticky. They stick with each other, and they form a network or mesh at the wound. That is your clot. Now blood cells are trapped in the network of the fibrin. So it is a network, okay. So red blood cells, white blood cells will not be allowed to flow out. The network then shrinks. It becomes hard, if you have noticed, and squeezes out the rest of the plasma, which is in the form of a clear liquid. That is the serum. That is the liquid part of blood will come out, and it will move out from the clot as a clear liquid. The clear liquid is known as the serum, and the solid mass which is left behind is called a clot or a thrombus, okay. So here you can see diagrammatic representation. A vessel is damaged here, okay. Red blood cells and platelets are there, blood vessel is there. So the platelets come here, disintegrate, okay, and then finally what is formed? Fibrin is formed, and red blood cells, white blood cells will get trapped here, and the clot is formed.